Okay, I think that I am going to get going at this point. It's really good to see all of you and for this day three of the workshop. Not to downplay the first 2 days, but they were really meant to be, getting people up to speed and acquainting you with the resources on NMR box and NAN and logging into spectrometers and a little bit of NMR training. But today is really where the main goal of the workshop is starting. So as I've talked about yesterday in my introduction to NMR, we have three general areas that sound fairly unrelated, but in my mind, they're quite related to each other. But today is this um, approach of taking 1D NMR metabolomics data, proton NMR data, and applying this technique that we're going to hear about from Frank called SAND. And that for us, for our lab, has really changed everything. We participated with Frank on the development of this, and we've been sent. We get it produces such nice results that we've been essentially applying it to most things that we do. It really changes the way we can think about a metabolomic study. So I'm really happy that you'll be able to hear from Frank himself about this. So without further ado, I just want to introduce Frank by saying if any of you have been involved in NMR at all for the last more than a few years or so, you will know Frank Delaglio, and you probably have referenced Frank Delaglio several times because Frank is he has been one of the key members of the NMR community for many years. He really came onto the scene, I would say. He was doing things before this, but I think I really first became aware of Frank when he was in Ad Bax's lab at the NIH. And I think it was at that time that he put together and published NMRPipe, which is the most widely used NMR processing package out there. It's been my mainstay for NMR processing for many years. It's available for no charge to people. You can either download software, or the thing that really has made it a lot easier for many of us is just to use NMRPipe on NMR box, which works great. And so, but Frank has really really changed the way we can approach NMR data by his contribution to NMRPipe. He's done much more than NMRPipe. But he remains the real, I think, the recognized leader in the field in understanding the nuances of data processing, writing code that gives incredibly robust data processing results. And I think that we wouldn't be where we are as a field today if it were not for Frank's contributions. For the last several years, I'm trying to think of how many, probably 6, 7, 8, 10. Frank has been at NIST. How long have you been at NIST, Frank? You're still muted right now.
Almost 10 years now.
10 years, my gosh. Frank has a position at the National Institutes of Standards and Technology, the measurement agency with our government, and he runs a lab where they are doing all sorts of interesting applications, including looking at antibody proteins that are modified in production for biosimilars, and he's working on real-time measurements of metabolomics, but he still is really at his heart he is a truly amazing NMR data processor. And so we've had the good fortune over the last few years of working with Frank. It all started by me. It wasn't a complaint, but I reach out to Frank periodically, pretty regularly, with like, hey, Frank, here's a spectrum. I don't know I can't process it very well. Can you help? And one of the last ones that I did that before we started working together on this time domain work was something that had baseline problems. And he said, the right way to tackle this is really to do time domain analysis. And so then we spent a year or more working on this. It was really fun, and I learned a lot. And so anyway, I'm happy to turn it over to Frank, and he will share his slides. I should say I just uploaded Frank's slides to day 3 on NMR Box, and you will get them in your events folder sometime during his presentation. They should show up there, but they probably won't be there quite yet. Okay, Frank, take it away.
All right. So thank you very much indeed for that remarkable introduction. The main reason that the software and methods that we've developed have been so impactful is that we've had the input and collaboration of so many fantastic scientists, many of whom are on the call right now. So thank you to all for the contributions to the work that we're going to see. So to introduce myself just a little bit, I'm speaking to you from the Institute of Bioscience and Biotechnology Research. It's a small institute in the Washington, D.C. area, which is jointly run by the University of Maryland and NIST, the National Institutes of Standards and Technology. And I'm part of a circle of groups that develops measurement methods to support biomanufacturing. Now, for us in the past, biomanufacturing primarily meant the manufacturing of monoclonal antibody drugs. And this class of drugs is the fastest growing class of drugs. And the reason for this is that since an individual can generate about 10 billion different variations on an antibody, it's relatively easy to find an antibody that binds well and specifically to most any kind of target. And importantly, antibodies of the same class are very similar to each other in chemical behavior, so that the monoclonal antibody serves as a general purpose platform for drug development, and the methods to develop them, test them, deliver them are very similar across the antibody class. And this makes them very attractive platforms for drug discovery. Now, monoclonal antibodies have to be expressed by live cells, generally mammalian cells. And so these two things have led us at NIST, where we like to develop measurement methods and reference materials, to develop two important reference materials. One is the NIST mAb, a monoclonal antibody reference material. And then more recently, the NIST CHO, a CHO cell that has been engineered to express the NIST mAb in pharmaceutically relevant qualities. And so these two together are a test bed for practical biomanufacturing and measurement techniques. And we're interested to characterize the kinds of bioreactions that are used to produce these antibody medicines. And so we want to take systems like the NIST CHO cell and study their metabolomics during cell growth. And that's how the groups here became involved in metabolomics studies. What we'll do in this presentation is talk about some of the mathematical details of NMR signals to try to explain why we're interested specifically to try to model them in the time domain and what advantages we expect to gain by modeling spectral signals in the time domain. And then we're going to explain how the SAN method works, how we do this time domain modeling to get such excellent results, as we've heard from Professor Edison. So in your introductions, I think you've seen already that in an NMR experiment, we measure a time domain signal. And in the ideal mathematical form of these signals, they are exponentially decaying sinusoids. And we use a method called the Fourier transform to take this time domain data and turn it into a frequency domain. And so In the time domain, we have exponentially decaying signals that extend throughout the data, and the Fourier transform tends to concentrate those signals into particular regions that we call peaks. The information content in both of these domains, in the time domain and the frequency domain, is more or less equivalent, and we can go backwards and forwards at will, but with a forward transform or inverse Fourier transform. So, to some extent, the process of converting this time domain data into a spectrum is really for the benefit of visual analysis, because people can look at a spectrum like this and make decisions, but they can't so easily look at time domain data like this and make decisions. Now, in a directly detected experiment, like a 1D proton experiment, we have some radio frequency pulse in the pulse sequence, and then we listen with a coil that kind of serves as an antenna to the radio frequencies that are emitted by the sample. And as you've seen, we Fourier transform these to get a spectrum. We digitally sample this information at uniformly spaced time steps. In a two-dimensional experiment, somewhere in our pulse sequence, we have a pair of pulses with a delay time between them. And we systematically increment this delay time between the pulses. And at each one of these delay times, we acquire another one-dimensional experiment. Okay. So what this does in practice is it takes the signals that we can see in any one-dimensional experiment, and it modulates them in an indirect dimension according to some chemical information that we program into the pulse sequences. And so what this gives us is a stack of 1D spectra that form the time domain of our two-dimensional experiment, and we Fourier transform all the rows of this time domain data to generate an interferogram. And then we Fourier transform all the columns of this interferogram to generate a two-dimensional spectrum. And what you can see in the interferogram is that each one of these 1D slices has the same signal in it, but that signal is modulated in intensity across the experiment. So it looks like a time domain signal in this indirect dimension, so that when we Fourier transform it, we get a spectral signal in that dimension too. Now, these two-dimensional spectra, we could think of them as surfaces like mountains. And so we can visualize them in 3D, like you see here. And what we talk about as peaks actually look like little mountain peaks. And this is a very nice way to get a qualitative understanding of the spectral data. But for practical analysis, this is not a very handy way to visualize spectra, because we want to do things like identify specific locations of peaks and make decisions about whether two peaks are lined up in one dimension or another. So in practice, we display two-dimensional spectra as contour plots, like the ones that we see here. And because of the fact that the underlying signals are 1D signals that are modulated in another dimension, all the signals in an NMR spectrum have this underlying form where a two-dimensional signal is actually expressed as the product of two one-dimensional shapes. And for time domain data, those one-dimensional shapes are time domain profiles, exponentially decaying sinusoids in the ideal case. When we Fourier transform all the rows, this still holds, but now the shape in the Fourier transform dimension is a spectral line shape, a Lorentzian in the ideal case. And then finally, when we Fourier transform both dimensions, that signal can be described as the product of these two one-dimensional line shapes. So These specific details of the NMR spectrum's mathematical form give us a way to handle NMR data in order to do different analysis tasks that would be difficult to do in other cases. And sometimes it'll be easier to do a task on time domain data. Sometimes it'll be easier to do a task on the spectrum. And in some cases, we want to do the task in the interferogram. So we're going to see some different cases and why we might do this in one way. Now, here's an example of a 1D proton spectrum that was measured by Chris Hesselman, who's with us on the call. And as you've seen in the discussion, each one of these peaks in the spectrum is due to a hydrogen atom somewhere in a molecule in this mixture. Okay. In a two-dimensional version of this experiment, we measure many different versions of this 1D spectrum where the intensity of the peaks is modulated by interesting chemical information. For example, we can make a two-dimensional experiment that we call a two-dimensional heteronuclear single quantum experiment. And in this experiment, we modulate each hydrogen signal by the frequency of the carbon atom that signal, that atom is attached to. So each one of these peaks in this 2D spectrum matches up with one of the peaks in the 1D proton spectrum, and the coordinates of that peak in the two dimensions are the NMR frequency of the hydrogen atom that's in the horizontal dimension here, and the carbon frequency of the carbon atom that it's bonded to. And the positions of all of these peaks is characteristic of the underlying molecular shapes and the chemical environment that they're in. So these chemical shifts, the positions of these peaks, are influenced, of course, by the molecular geometry of the molecules, but also by the nature of the solvent, the pH, the ionic strength, the temperature, and the dynamics of the underlying molecule. So these spectra encode all of this information at once. And so they're an incredibly rich source of understanding about molecular processes. Now, these multidimensional experiments are time-consuming because we have to measure a whole series of individual one-dimensional NMR spectra. And so one of the tools that we can use to potentially decrease the measurement time of a two-dimensional sample is a method called non-uniform sampling. And in the case of metabolomics, which, as you've seen already, we often want to measure in a time series as quickly as possible to get time-resolved data. The amount of time it takes to make a measurement is critical. So this non-uniform sampling method is often of great importance when we want to measure two-dimensional spectra on metabolomic samples. And the way in which this method works is rather simple. In an ordinary acquisition, we, as have mentioned, have a time increment between two radio frequency pulses that's incremented in a systematic way, so that we would generate a series of 1D measurements, and the time it takes to make the collection of 1D measurements is just based on the number of individual 1D measurements that we make. In non-uniform sampling, we randomly skip some fraction of the underlying measurements. And since we're making fewer 1D measurements, the experimental measurement time is less. The challenge is that when we have data of this type, when we use the ordinary Fourier transform scheme, we Fourier transform all the rows and get individual spectra, that's fine. But when we Fourier transform all the columns, since there are gaps in the spectrum or gaps in the data, we get something that looks like a spectrum, but it also has these wild artifacts. And to understand where those artifacts come from and what we can do about them, we have to understand a little bit about the Fourier transform. The Fourier transform, as we use it on experimental NMR data, looks like this. Little x of t is our NMR time domain data, and big X of f is our corresponding spectrum. And to generate one point in the spectrum, we multiply our time domain data by an exponential with an imaginary term here. And then we add up all the points of that product, and that forms one point in the spectrum. This exponential can be decomposed or described in terms of a real part that's a cosine term and an imaginary part that's a sine term. So in practice, all we do in a Fourier transform is take one of these Fourier terms, which is complex, and the real part is cosine, and the imaginary part is sine, and we multiply it by our time domain data, and that forms a product, and we add up all the points in that product to generate one point in the spectrum. And it's only in situations where the frequency of the Fourier term matches one of the frequencies in the time domain data that we get a product that's either predominantly positive or predominantly negative. So when we add up all the points, we get a peak in the spectrum. Now, this nice behavior only holds true when we have a continuous signal. And so in practice, when we have a shorter signal or a fewer number of points, the kind of result that we get from the Fourier transform is no longer optimal. So in a high resolution 1D spectrum, we might measure 16,000 points, and we get a corresponding spectrum that look like this. But as we measure fewer and fewer points and get data that's more and more truncated, the corresponding Fourier line shape becomes less and less ideal. It becomes artificially broad, and it has these regular ripples that we call truncation wiggles. And so what that means is the corresponding spectrum that we get from Fourier analysis, Fourier transforms, depends on how we did the spectral processing. So all of these spectra that you see here all came from the same underlying signal, just sampled in a different way. And this gives us one of the motivations as to why we would want to model data in the time domain instead of in the frequency domain, because in the frequency domain, we have to account for whatever spectral processing was applied to the data. Now, in the case of an ordinary measurement where we can only measure a limited number of points, as in the carbon dimension of a 2D experiment, we get data that's truncated, and when we Fourier transform data like this, we get an artificially broad signal with these regular truncation wiggles. And in the case of non-uniform sampling, where instead of having the gap at the end of the data, we have little gaps inside of the data, when we Fourier transform data like this, we get a nice sharp signal, but we get what are apparently noise-like artifacts that look random. They're not random, though. They are completely determined by the pattern of points that we kept and the pattern of points that we skipped when our non-uniform sampling measurement was made. In both of these cases, we have two different sampling schedules. They both have artifacts associated with them. For this uniform sampling schedule, the artifacts are oscillating and random. And for this non-uniform sampling schedule, the artifacts sort of look like random noise, but they're not random. Every peak in a spectrum like this will have the same artifacts. and the size of the artifacts associated with the peak are proportional to the size of that peak. This actually gives us a way to reconstruct the data and minimize the effect of these artifacts. And the method that we can use to do that is called iterative soft thresholding. So we're going to take advantage of the fact that every peak in the spectrum has proportionally sized artifacts, and the artifacts are the same. The shape of the artifacts are the same for all the peaks. And the methods that we use are pretty simple. We draw a threshold that's relatively high, and we transfer some of the intensity from the top of the largest peaks to a secondary spectrum. And then we inverse transform this data back into the time domain, delete the points that were skipped during the non-uniform sampling, and then transform it back into the frequency domain. And we can repeat this over and over again, making the spectral signals smaller and smaller by transferring some of the intensity from the tops of the peaks to a spectrum that we accumulate. And as the signals in the original data get smaller, the corresponding artifacts get smaller too. So in effect, this method is filling in the missing spaces in our data. And it fills in the missing spaces in the interior of the data or at the end. And using this method enables us to go from a non-uniformly sampled spectrum that looks like this when it's processed with a regular Fourier transform to a non-uniform sampled spectra that looks like this when it's processed with IST. And for all intents and purposes, this spectrum is comparable for analysis purposes to the fully sampled data. Now, something else that's important to point out and to help us understand why we want to model data in the time domain is that the Fourier transform, the tool that we use to move from time domain to frequency domain, and the inverse Fourier transform that we use to move from the frequency domain back to the time domain, those two transforms are essentially the same. Their Fourier terms both have a cosine real part and a sine imaginary part. And so what that means is the rules that relate one domain to the other work in both directions. And so very specifically, the two things to keep in mind is that the position of a point in one domain represents the frequency of a sinusoid in the other domain, and the amplitude of a point in one domain represents the amplitude of the corresponding sinusoid in the other domain. So what does that mean for us in practice? Well, when we consider time domain data, as we've said, each point in the time domain data means a sinusoid in the spectrum, in the frequency domain. And the first point in the time domain data is a zero frequency sinusoid, so just an offset. The second point in the time domain data is one cycle of a sinusoid. The third point in the time domain data is two cycles of a sinusoid in the corresponding spectrum. And so what that means in practice is for cases where a spectrum has baseline distortions, like the one that you see here, and this is a carbon spectrum that was measured there at UGA on a special carbon probe that was designed and built there. This complicated baseline is really due to just a small number of points at the beginning of the time domain data. And if we can use a mathematical method like IST to replace 5 or 10 points at the beginning of the time domain data, we can remove this baseline distortion. And that's what's shown here. So by modeling in the time domain, we can avoid the problems that are associated with baseline curvature in the frequency domain. So that leads us now to the discussion of SAND, the method that was developed by Yue Wu and Omid Sanity there at UGA, with help from lots of other folks. And this has been published fairly recently in the Journal of Analytical Chemistry. It took us about half a year of duking it out with reviewers. Well worth the effort. Now, SAND, our time domain modeling method, which we're going to discuss in more detail, is a kind of follow-on to a very successful and similar time domain method called CRAFT that was published by Krish Krishnamurthy, and it's based upon a Bayesian analysis program that was developed by Larry Bretthorst, who is a genius of Bayesian mathematics, a foundational contributor to Bayesian statistics. And this Bayesian module for modeling time domain data by exponentially decaying sinusoids is about as good as it could be. It really can't be improved upon for this particular application. And so CRAFT, which uses this as its internal engine, has been remarkably successful. But this underlying Bayesian tool is proprietary. The code is written in Fortran, and it's specifically dedicated to 1D data that are exponentially decaying sinusoids. And it can't be adopted to other models and to multidimensional data or to non-uniform sampling. And that gave us the motivation to try to come up with an alternative to it. And just to show you why we need alternatives, what we see here is a portion of an experimental spectrum that's modeled in the time domain with ideal exponentially decaying samples. So the model is shown in yellow, the measured data is in blue, and the residual between the two is shown in red. Okay, so what you will see is that there are systematic differences between the measured NMR data and the ideal model. And in this case, those differences are much, much bigger than the random noise in the spectrum, which is almost not noticeable at this. Now, if we change the model to, say, a Gaussian decaying signal, we actually get a slight improvement in the agreement, but still a very substantial residual between the model and measured data. What that means is we need to use more elaborate models. So, for example, here are some different cases, including at the end here, models where the phase of each peak is allowed to vary individually, and each peak is modeled in terms of the sum of an exponential term and a Gaussian term, each with its own amplitude, and an unresolved cosine modulation, so an unresolved coupling built into the peaks. And so we can make these models more physically realistic, but in order to do that, we need a time domain modeling technique that we can access and modify ourselves, and one that isn't hard-coded to work only with one-dimensional exponentially decaying sinusoids. And a final motivation is that if we wanted to make models like this in the frequency domain, we would have to accommodate all the details of Fourier processing. But in the time domain, once we have a model for a time domain signal, we don't have to change that model at all to put it in the context of non-uniformly sampled data, because the underlying time domain signal is the same in all of these cases, whereas the corresponding spectrum that we see is different in all of these cases. So by modeling in the time domain, we can adopt our modeling technologies, whatever method that we use, to be able to analyze non-uniformly sampled data as well. And that's a long-term goal of the work that we're doing. So how does the SAND method in its current form work? Well, the Kraft method uses Bayesian statistics to decide how many peaks should be used in order to model a particular region of the spectrum. SAND uses a procedure that's commonly used in machine learning, which is to take the data and divide it between a training set and a validation set. And this is a method that we use to avoid overfitting of the data. And the idea is that we take the randomly selected points that are part of the training set and use those to model the signal. And we try modeling a spectral region with one signal, with two signals, with three signals, with four signals. And of course, each time we add a new signal, the improvement between the model and the experimental data gets better. But that doesn't necessarily mean that the model is more appropriate. So we could add 100 signals and match the experimental data perfectly in a given region, but the underlying values that we got for those 100 signals wouldn't be meaningful. So what we do in the SAND approach is fit the data with the training set, and every time we propose a new peak, we perform the spectral model and see if the model that we fit to the training data improves the agreement to the validation data that was never used in the fitting procedure. And it's when the improvement to this validation set stops improving that we know we can stop adding new signals to the SAND model. So that's what we do. We start with a spectrum. We subdivide it into regions that are easier to handle because they have smaller number of peaks. We transform that into the time domain, split the data into a population of training points and validation points. And for a given region now, we optimize parameters for one signal, optimize parameters for two, for three, and so on, and use the testing of agreement with the validation data as a way to decide when we've added enough model signals to describe a particular spectral region. And when all that's done, we get the parameters of the model in terms of the frequency of the underlying sinusoids, the exponential decay of the underlying sinusoids, and the amplitude. Okay? And it was our goal to try to set this up in a way that required no user interaction to perform and didn't need a graphical interface in order to do this analysis, which means that we can conveniently apply it to batch and analyze data, and we could apply it in a way that didn't depend on the judgment of an operator in order to get the results. The bucketing scheme that was used to divide the spectrum up into analysis regions is an adaptive bucketing scheme that was published by others, whereas conventional bucketing would generally be uniformly spaced. We can use adaptive bucketing to try to make sure that each spectral region has at least one or more peaks. And the SAND algorithm basically is what we've described. We have a time domain model for the peaks, which in this first version of SAND is a simple exponentially decaying sinusoid. We divide the time domain data into a training set and a validation set. And because the optimization method that we use is in part random, we do the optimization 3 times. So we do a SAND analysis 3 times repeatedly on the same region to make sure that all the values are converged properly. And what we do in this analysis is, for a particular region, we scan through each possible frequency position in that region and find out where the largest signal is likely to be. So we insert that signal and optimize its parameters by basically random coin flipping that we call Markov chain Monte Carlo method. And then every once in a while, we pick a particular signal and parameter and optimize its parameters in a systematic way, in this case, with a method called fixed point optimization. And that procedure looks like this. We choose a peak at random and one of its 4 possible parameters. We change that parameter by a small amount, and we test whether that change makes the fit better or worse. If it makes the fit better, we keep it. If it makes the fit worse, we keep it in a probabilistic way. We toss a coin, and the coin tells us whether we should keep the change that makes the model worse or reject it. And we tune that probability in order to accept a small number of results that make a model worse as a way to jump out of local minima in this very complicated optimization problem. So Sand uses the Monte Carlo method called the Metropolis method, which is a special case of simulated annealing that maybe we'll describe the next time we all get together. So how does SAND perform? Here first are some tests on simulated data. And in this set of simulated data series, we have 2 broad peaks and 1 narrow peak. And in this series, the narrow peak moves from this position, where it's clearly separated from these 2 broad peaks, to gradually getting closer and closer, and then even more or less within one of these broad peaks. And in 1 or 2 of these series, the small signal is so far into the interior of the larger signal that it can't even really be seen visually. So a human analyst might have a problem identifying this small signal in the presence of a bigger, broader one. But what's exciting to notice is these are the estimations that come out by SAND, and SAND is able to identify this smaller peak and its correct chemical shift to within about 0.04 amplitude. So that's kind of remarkable. Now, to get into a more realistic example that matches what you would see in a metabolomics spectra, these are simulated spectra that were simulated to have peaks in many different locations and at several different line widths to kind of mimic what you might see in a metabolomics spectrum. And one of the challenges with this kind of work is that for a very complicated spectrum, there might not actually be enough statistical information to make an unambiguous decision about whether a particular region is 1 signal or 2 signals or 3 signals. So one thing that's common, regardless of whether an automated method or a human-based inspection method is used, that there will be cases where a peak that is known to be a single signal will be decomposed into more than 1 signal. And so here's a case where that happened. Here's another case where this peak here was decomposed into 3 smaller peaks. So this is expected in spectra where there are several hundred or several thousand overlapping peaks. And this issue makes it very difficult to compare the effectiveness of one optimization method over another. So as a way to make this comparison and this judgment about how effective the modeling is, to make this more tenable, we took a collection of urine spectra, and these urine samples have added to them different amounts of a 4-compound mixture. And so since we know the signals in the 4-compound mixture, we can relatively easily identify those signals within the complicated urine spectra and track the intensities of those peaks. And what we expect is that the intensities of peaks that belong to the spike-in compounds should increase linearly with the amount of spike-in mixture that we add to the urine. And so, in fact, at the little graphs at the top, you can see the integrals of particular peaks in given regions where we know there are compound spike peaks. And in all the cases, we get remarkable correlation between the amplitudes that we measure in the spectrum and the expected linear behavior with the amount of spike-in compound. So this is how we validated SAND. In order to make this as convenient as possible to use, we implemented it inside NMRPipe in a tool called sand1d.com, which you'll see later, which is a way that you can name a spectrum and an analysis region and leave it alone and come back and get a result. So here's an example from the data that we've shown. The measured spectrum's in blue, the SAND model is in yellow, and the individual model components of the SAND model are shown here. No interaction was needed, just name the spectrum, the analysis region, and we get a SAND result. This is what we were aiming for. Now, this is done with the underlying tools of NMRPipe, which is a pipeline-based processing program. And this is an idea from the Unix operating system, the idea of a filter program that takes some kind of data, reads it in, processes it in some way, and writes it out. So, for example, a Fourier transform filter that can read a series of data vectors from time domain NMR data and produce the corresponding spectra as output. And this leads to the idea of a pipeline, which is just a collection of these programs connected together in series, each performing a different step of the spectral processing, like a digital filter or what we call a window function or apodization, and Fourier transform, and so on. And all of these things can be assembled into Unix shell scripts that look something like this. Now, this is terrifying to anyone who has not seen a Unix shell script. And the learning curve for building these shell scripts by hand is very steep. So we've done two things. One is we have a series of commands that can automatically generate an elaborate processing script. And then to actually use these, we have a graphical interface that lets you graphically specify processing parameters and uses these underlying commands to automatically generate and execute processing schemes. And we'll see examples of those. And then the slides have some notes about how to use these processing panels for uniformly sampled data and non-uniformly sampled data, and perhaps we'll see some examples today. But then also there's a second graphical interface in this software called SpecView, which is specifically for viewing 1D and 2D spectral series. And the main purpose of this interface is to be able to look at a spectral series and quickly perform principal component analysis on a region. So in this interface, highlight a spectral region, press a button, get principal component analysis, and you can click on any of these spectra and see which point in the PCA scatter plot it is, or click on something in the scatter plot and see what the corresponding spectra is. So this lets us get a very quick overview of a spectral series and figure out why different spectra behave differently. Okay, so that's an overview of my presentation for today. And by the way, if any of this stuff was of interest to folks, we always have postdoctoral opportunities here at IBBR. So that is the presentation. Thanks for your patience in many different time zones there. Thanks for joining in.